This is lecture 14, Neurophysiology. Begin a discussion of sensory physiology, focusing on sensory receptors, sensory receptor potential, and then overview of the peripheral nervous system and neural integration. This is a slide we had seen previously, looking at the general structure of the nervous system within organisms. With a sensory input, we are going to, of course, talk about the receptor interface, generating that initial sensory signal that's carried to the integration area, the central nervous system. We'll look at how this information is integrated throughout the central nervous system. And then, of course, the motor output, targeting the effector that will become the focus of future lectures on reflex arcs and the neuromuscular function with focus on muscle physiology. First, the overview of the peripheral nervous system. We begin the discussion with sensory receptors, so the origin of the information coming from either external or internal environment. Let me set the arrow here. The sensory receptors are classified by the stimulus type that is detected. The mechanoreceptors, thermoreceptors, of course, mechanical distortion, temperature, photoreceptors, obviously photons of light, chemoreceptors, a variety of chemical signals that can be picked up. We saw examples of chemicals being able to affect cells in discussions of the humoral type of regulation of endocrine cells. This will be somewhat different, but we will see that it's still quite similar. And then nociceptors, those picking up pain signals, so threat of harm. This table shows the characteristics of the sensory receptors. The left column is the receptor class. The middle column is the sensation or visceral information, and the right column is the modality. First, we see the photoreceptors, obviously vision and photons of light. Then the chemoreceptors, taste, smell, pain, blood oxygen, blood pH, and of course this isn't all types of pain, it's the types of pains that might be caused by a caustic agent. So chemicals dissolved in the saliva, obviously for taste, chemicals dissolved in the mucus for smell, chemicals in the extracellular fluid. If you've ever gotten a shot where there's something in it that causes it to sting, that's the type of receptor we're thinking about here, or bit by an insect, say a fire ant for sure. Oxygen dissolved in the plasma for the blood oxygen, and then the free hydrogen ions in plasma for sensing of blood pH. We will see when we talk about the respiratory system and pH balance that this index is used by cells also to get an indication of the levels of carbon dioxide. So the organisms have capabilities of sensing blood oxygen and carbon dioxide levels. Of course, whatever's in the blood is similar, if not completely identical, to the interstitial fluid. Thermoreceptors are of two types, the warm and the cold. So warm receptors detect warmth. So there's an increase in temperature. Temperatures between 37 and 45 degrees. And cold receptors are going to detect decreases in temperatures between 37 and 20 degrees Celsius. The mechanoreceptors, there's baroreceptor for blood pressure, osmoreceptor for the osmolarity of the extracellular fluid, and then the hair cells for sound and balance and equilibrium. So blood pressure is detected by baroreceptors through a stretch of specific blood vessel walls that are selectively innervated by these types of sensory neurons, and the swelling or stretch of receptor cells as they are affected by the osmolarity of the extracellular fluid. Obviously, sound waves and acceleration are going to impact the hair cells for the vestibulocochlear system. The classification by location is a slightly different approach. So those that are capable of picking up external environmental cues are exteroreceptors. Those for internal, mostly visceral types, are interoreceptors. And then those that are basically picking up position from the musculoskeletal system are the proprioceptors. This table is showing us the general sensory receptors classified by the structure and function. And we see the anatomical class or structure in the top left, the left column here. And then in the center, an illustration of these, the second panel column. The functional classes according to location and stimulus, L for location, S for stimulus. And then the body location. We start first then with the uncapsulated versus, of course, the encapsulated. The uncapsulated includes the free dendritic nerve endings of sensory neurons, the modified free dendritic endings such as Merkel's discs, or the root hair plexuses. The encapsulated includes Meissner's corpuscles and Krauss's end bulb. Let's continue across for each of these individually then. The free dendritic nerve endings of the sensory neurons, we see, for example, here, multi-layered epithelial lining. Very likely this would be the skin, the epidermis. And we see the free nerve ending distributed among the cells at this level. And the first level of myelination begins just deep to the deepest layer of cells, of course, within the underlying connective tissue, so epidermis and dermis here. So these are obviously exteroreceptors. They can also be interoreceptors, if, for example, this might be a mucous membrane lining the oral cavity or the digestive system. Less likely the digestive system beyond there. The stimulus type is nociception, pain, thermoreceptors, heat and cold, and possible mechanoreceptors for pressure. The body location, most body tissues, most dense connective tissues like ligaments, tendons, dermis, joint capsules, periostea, and epithelia, such as the epidermis that I had mentioned, the cornea, mucosa, and glands. The modified free dendritic endings, the Merkel's disc, we see that there is a sensory cell interfacing with the nerve terminal in this case. These are characteristic exteroreceptors, predominantly the mechanoreceptors for light pressure, and they are slowly adapting. We'll look at this phenomenon as we proceed. So obviously we are now identifying light pressure, so there's going to be a dedicated sensory receptor system for deep pressure. This is found within the basal layer of the epidermis of the skin, so it would proceed no farther than just this basal layer. So this would be a free nerve ending in the surface of the skin for pain and temperature, and maybe mechanoreceptor, some pressure, but the Merkel's disc somewhere nearby would be picking up the light pressure. And then, of course, we all know that we can feel movement, say air moving across the hairs on our arms. these are the root hair plexuses. So here is the hair follicle, and at the base of the hair follicle, within the dermis, the nerve endings will be surrounding that hair follicle structure. So these are obviously exteroreceptors. They are mechanoreceptors for deflection, and they are rapidly adapting. So we know that we perceive this sensation initially, and then if it continues, we are able to put it on a lower level of priority as far as sensory information coming in. These are located in and surrounding hair follicles. Next then are the encapsulated, such as Meissner's corpuscle shown here. We'll look at the structures of these, maybe not all of them, but most of them as we proceed in more detail. Krauss's end bulb shown here. So in these two examples, there's going to be some other cellular structure associated with the nerve ending, and of course encapsulating the nerve ending. So unlike the hair follicle here, where the nerves come in and are coming in from the outside and surrounding it, these will have the nerve endings inside with a cellular structure completely surrounding it. So the Meissner's corpuscles are exteroceptors, to pronounce that correctly. Very typically, mechanoreceptors, also light pressure, discriminative touch, vibration of a low frequency. So these are the types of sensory receptors targeted by the classical tuning fork used by the physician or neurologist to study sensory function to a certain area of the body. The dermal papilla of the hairless skin is a prime location of these, the highest densities, particularly, of course, the nipples. We've discussed in neuroendocrinology the milk letdown reflex involving oxytocin. So these would be the types of mechanoreceptors picking up some of the sensory signal for that reflex. External genitalia, fingertips, soles of the feet, and the eyelids. Krauss's end bulb, also exteroceptors. These also are mechanoreceptors, so they're probably a modified Meissner's corpuscle. You can see very similar, just slightly different organization of the cellular capsule. These are found in the connective tissue of the mucosa, so for example the mouth and the conjunctiva of the eye, and of hairless skin near the body openings such as the lips. We continue to look at the general sensory receptors classified by structure and function, and are proceeding through another collection of the anatomical class, the Pacinian corpuscle, Ruffini's corpuscles, muscle spindle, and the Golgi tendon organs. The Pacinian corpuscle we see here is surrounded by an onion-shaped multicellular structure, so layered like an onion with a nerve ending terminating inside it. So whenever we see this, we know that these cells surrounding it are most likely going to change with the appropriate sensory stimulus and somehow cause a depolarization wave in the nerve ending, the peripheral process of the sensory neuron terminating within it. So the functional class, according to location and stimulus types, the Pacinian corpuscle is an exteroceptor, and interoceptors, and some proprioceptors will utilize this structure to pick up appropriate sensory information. They are mechanoreceptors. These are going to pick up the deep pressure, the stretch, the vibration of the high frequency, and they are rapidly adapting. The body location will be the subcutaneous tissue of the skin, the periostea surrounding bone, mesentery, tendons, ligaments, joint capsules, most abundant on the fingers and the soles of the feet, the external genitalia, and the nipples. So we're seeing that there are mixtures of these types of sensory receptors distributed through different regions of the body. Ruffini's corpuscle is an exteroceptor and also proprioceptor, and it is also a mechanoreceptor, but picking up deep pressure and stretch, and they are slowly adapting, so somewhat similar to the Pacinian corpuscle. However, that is rapidly adapting, and these will be the slowly adapting, so providing different types of information to the nervous system, a sustained source of information and one indicating immediate change. Of course, deep pressure is extremely important. Think of being bitten by a bear. This is critical information that has to be organized. Is it a quick bite, or did it persist? To take it to the extreme, I guess. Deep in the dermis, the hypodermis, and the joint capsules is where we're going to find the Ruffini's corpuscle. Muscle spindle, obviously in the skeletal muscle. We'll look at it in great detail, as well as the Golgi tendon organs when we cover the neuromuscular system. So the proprioreceptors and mechanoreceptor type for muscle stretch is what we will study relative to those. The Golgi tendon organ also for proprioception, but this is stretch at the tendon, so they're found in the tendons, obviously. And then there are also the joint kinesthetic receptors, the Pacinian and Ruffini corpuscles, free dendritic endings, and receptors resembling the Golgi tendon organs that collectively function as proprioceptors, and these are thereby mechanoreceptors and nociceptors in the joint capsules and of the synovial joints, the freely movable joints. So obviously in this integrated system that we're looking at, that we use to talk about the endocrine system and interaction with the nervous system, we are now layering on top of this a receptor interface with internal or external environment and an afferent signal being generated. So the afferent or sensory neuron is either acting as the receptor or conjointly with a cellular structure that is functioning as the receptor, but picking up the afferent signal and delivering it to the integrating center within the nervous system. The CNS was the model that we saw as the target of the afferent neuron. This will be integrated, of course, when needed. Call in the endocrine system. But the efferent signal that will be generated targets the effectors to bring about an appropriate response. This is another figure that we had seen before, and predominantly now what we're focusing on are the sensory receptors for the somatic or the voluntary nervous system, but there is a linkage with the involuntary or autonomic nervous system, as we are all aware. So there will be the sensory neuron, the afferents, that can also receive or are involved in picking up the enteric nervous system information for a visceral type of a reflex, so stretch of the gut tube versus sensory information, say vibration coming to the palm of the hand. So these are not the identical neurons that are doing this, but in this type of a model, it is the same general class of neurons, the afferent or sensory neurons. and these afferents carry it to the integrating center that includes the spinal cord, of the CNS, as well as the brain. Of course, information will have to ascend up through the CNS, either spinal cord or brainstem, to the brain to deliver that information to higher centers for integration, with contributing descending information to modulate or initiate or impact the the output through the efferent neurons, and our focus will in part be on the again the voluntary nervous system, the somatic neurons, but we are keeping in mind the visceral type of reflex responses that will control the differential activation and inactivation of the sympathetic and parasympathetic nervous systems that are controlling the smooth muscle, cardiac muscle, the exocrine glands or cells, as well as some endocrine glands and cells, and of course some adipose tissue. Here we have the impact of the enteric nervous system within the gut tube. Whenever you see enteric, you know that it's with reference to the gut tube, and that provides some control on those as well, integrated within all of this, because the autonomic nervous system communicates very heavily through the enteric nervous system. So some of our initial reflex studies will be associated with the visceral reflexes, and then there's a committed lecture, lecture 16, to the somatic reflex that is targeting the skeletal muscles. So we have looked at the classification in a couple of different ways. Now we can classify based upon structural complexity, either simple or complex. We'll talk about transduction by the sensory neurons with reference to receptor potentials and generator potentials. This slide is showing two panels. On the left, the skin and how it feels many sensations. So an illustration to reinforce the point of regional collections of multiple types of sensory receptors. And then the panel on the right is the sensory transduction in a series of stages or steps that we'll follow to see how the entire system is integrated. That first slide was a very simplistic way of viewing this functional circuitry. So here is a little piece of skin. The epidermis is shown above here with the dermis, and then, excuse me, this is the epidermal layer down to this portion here, and then the dermis shown below, just getting into the hypodermal layer. So we notice a sweat gland with its duct coming to the surface, a hair follicle with the hair extending beyond the epidermal layer. First, we see over here on the left the bare nerve endings, so the pain, itch, and temperature occurring there. We see the Pacinian corpuscle just at the junction of dermis and hypodermis. So this was that pressure, rapid adapting. The hair follicle endings that we had mentioned, illustrated here. Ruffini's corpuscle for touch and pressure here. Meissner's corpuscle shown here for the light touch. And then the Krauss end bulb also for touch. So all of these contributing to our touch sensation: light pressure, deep pressure, vibration, light touch, and movements of the hair follicles, pain and temperature, and all of things of that sort. Then let's proceed to this illustration. We see in the top the stimulus, a sensory cell that might have some type of receptor protein. So the stimulus activates a receptor protein by changing its conformation. So regardless of the stimulus type, it's somehow going to change a receptor protein, and that of course is going to impact ion channels. We have to change the potential across the membrane of the receptor cell, so there will be a receptor potential generated. So the distortion or the conformational change of the receptor protein, then that will open or close ion channels, causing potential changes across the receptor membrane. And then a small stimulus can be amplified to produce a large response because of the receptor potential that can feed in either as a generator potential, bringing about action potentials if we're right on the, this would be an example of a free nerve ending. But if there's a receptor cell interface, as in option B here, there will be neurotransmitter release related to exocytosis with the receptor potential. Then the postsynaptic cell will be our afferent neuron, and it generates action potentials, and then ultimately the action potentials stimulate the neurotransmitter release, thus transmitting the information to other parts of the nervous system. So this output of the action potentials may actually even be, and most commonly will be, within the CNS, for example, the spinal cord or the brainstem. Here's 3 examples to illustrate some of the points that we just made of either free nerve endings or interface with receptor cells, but also to introduce the idea of the receptive fields. So we have panel A, B, and C. In panel A, there is a sensory unit in which the receptors are specialized endings of the afferent neuron here. In panel B, a sensory unit in which the receptors are separate cells, each of which communicates to a single afferent neuron. That's not what the illustration is accurately showing, but at least to a terminal of a single afferent neuron. And then in panel C, the receptive field for a sensory unit is depicted. So a classical way of describing this branching pattern of the axon is called the telodendria. So there are multiple branches of the axon. Excuse me, this is the peripheral process, sorry about that, but the same term applies. This would be the central process. So the peripheral process has this branching, profuse branching. It may or may not, but typically it has some. And the area where we find those distributed, however they are interfaced with the receptor function, it refers to the receptive field. So any sensory stimuli coming into this area, say of the skin, will be in the field where this cell can receive it. So it's the receptive field of that primary afferent neuron. So this would be the receptive field for this represented primary afferent or sensory neuron. And we see that the axon, the peripheral process branches, and so multiple cells will have to receive input, sensory stimuli, transduce that to an exocytotic event, and then there'd have to be postsynaptic receptors on the peripheral process terminals of the afferent neuron such that a depolarizing postsynaptic potential would be generated in EPSP or maybe in IPSP, as we'll see. But that will pass away as the graded potential, either a depolarizing wave for an EPSP or a hyperpolarizing wave for an IPSP. and eventually reach a spike zone and determine the firing rate of this sensory neuron. Of course, the free nerve endings, the receptor protein is expressed right on the surface of the peripheral process terminals and will be directly affected. Excuse me. Now this slide shows the example of the interface of the sensory cells with the primary afferent neuron. So these will be the terminals of the peripheral processes, as we're seeing here. So this figure is showing the structure and function of sensory receptors. In panel A, a sensory receptor that is specialized ending of an afferent neuron. The stimulus acts on the sensory receptor by opening or closing ion channels and thus producing a receptor potential. In panel B, a sensory receptor that is a separate cell from the afferent neuron. The stimulus changes the membrane potential of the receptor cell, which opens or closes a calcium channel, and a cytosolic calcium concentration increases or decreases, and the changes in the calcium concentration trigger or inhibit the release of a chemical transmitter by exocytosis. The transmitter communicates to the afferent neuron by binding to receptors on the free nerve ending. So let's look at these illustrations. So some mechanical deformation of this multilayered cellular structure is going to cause the depolarization wave that will spread to the trigger zone, where then there would be an action potential generated on this peripheral process. In this example, the sensory receptor protein is coupled to a calcium channel, a voltage-gated calcium channel. So there'll be an ionic event in the membrane that creates the voltage changed either to open or close this population of calcium channels. If the intracellular calcium increases sufficiently, it'll trigger the exocytosis, and then it's a classical type of paracrine effect of this neurotransmitter on postsynaptic receptors expressed on the surface of the nerve ending. Let's follow through with the example of the Pacinian corpuscle that we saw on the previous slide. So we can see the nerve ending coming into this and the multiple layers of cells. So it really looks like an onion. This is a section through it. So when you look at the surface of an onion, you don't see all the layers, but when you slice through it, you do. The Pacinian corpuscle within the skin is located deep within the dermis, typically close to the junction with the hypodermis. So this figure is showing the sensory reception in the human skin. Again, the epidermis, dermis, and hypodermis, with the Ruffini corpuscle picking up pressure, the free nerve ending with the pain, the hair follicle receptor for hair displacement, Meissner's corpuscle for pressure, and the Merkel disc for touch and pressure, and the Pacinian corpuscle is the focus here. So the membrane of the Pacinian corpuscle is illustrated in panel C, and we see that pressure is going to cause a distortion of the membrane, and that distortion of the membrane will actually open the sodium ion channel, causing a depolarization of the these cells, and that will be transduced to the excuse me, in looking at the description for panel C, the mechanical forces sensed by the receptors are thought to cause the sodium channels to open, leading to depolarization of the sensory neurons. So this may actually be located within the terminal of the neuron, and the distortion and interaction with the Pacinian corpuscle cells leads to depolarization of the terminal, and then a depolarization wave would pass from there to the spike zone and impact the frequency of firing of the neuron. This figure is showing the sensory cell membrane receptor proteins that respond to stimuli. So first we see the mechanoreceptor, then thermoreceptor, then electroreceptor, chemoreceptor, and photoreceptor as examples. So the mechanoreceptor is what we just saw. There's pressure with deformation, and it actually physically opens the sodium channel, allowing sodium influx and a depolarization at the membrane, as we are familiar with. The thermoreceptor, there's an enzyme that is temperature sensitive. So depending on which temperature direction it is sensitive to, say if this is for heat, as heat increases above basal body temperature, the 37 degrees Celsius, or the classical 98.6 that we're familiar with, then this enzyme activity increases, and that increased enzyme activity is going to open the channel and allow an ion to flow. The electroreceptor, an electrical charge opens the ion channel, so somehow there has to be a voltage change to bring about the voltage gated opening of the channel. In this case, it's a sodium channel that's being illustrated. For the chemoreceptor, such as taste or smell, the molecule binds to a receptor, initiating a signal that controls the ion channel via intracellular messenger signaling cascade. So here we see the taste or the smell molecule. It'll bind to an integral protein exposed to the external surface of the membrane, and many of these, at least, if not all, are G-coupled proteins, but other types of coupling can occur, and it brings about activation of the channel through a metabotropic type of mechanism. So it could be a sodium or potassium channel. Sodium is going to depolarize, and potassium, of course, is going to efflux and hyperpolarize. And then the photoreceptor, the membrane protein, actually is sensitive to photons, and in this example there's a cyclic GMP-mediated sodium channel, so the coupling activates an enzyme that's going to cyclize GTP into GMP. So we see another example of a nucleotide that can be altered from the triphosphate state to the cyclized monophosphate structure and serve as a second messenger. Well, we had talked about graded potentials from the perspective of postsynaptic potential. Now let's think of it as a generator or a receptor potential. It might be similar in properties to the postsynaptic potential, but we have a separate description for it because of the way that it is initiated. So it occurs in the sensory receptors and the sensory neurons when mechanically gated channels open. Here we see such an example where the muscle is the source of the information. I don't like that they're showing a bipolar neuron here because it should be pseudo-unipolar, as we had seen before, and therefore their illustration of the location of this recording should be way out here, and this would be the spike zone, and this would be the central or even the peripheral process beyond the spike zone. So this anatomical illustration is wrong. We know enough of the anatomy of sensory neurons already to correct this, but disregard that and still think of recording at the sensory terminal, a short distance away, where it will be the typical type of graded membrane potential approaching the spike zone, and then the spike zone, and then the action potential proceeding through. We'll see another illustration that corrects the anatomy on this and the location of the recording electrodes in a moment, but that's fine. We know enough already to make those corrections, but that does not change the nature of the event that will occur. First thing we notice at the sensory receptor terminal, the dendrites or peripheral process of the stretch receptor neuron, that the stretching a muscle stimulates the opening of ion channels in the stretch receptor dendrites, and this produces nearly a perfect square wave. When the stretch occurs, we get a change in membrane potential. We would almost predict that it would be sodium channels because of the direction of the change in the membrane voltage, membrane potential. That positive deflection almost always reminds us of a sodium influx. Doesn't always have to be, but this would be a typical case, as we saw from the previous figures. And then as that as that receptor potential migrates away from its origin down the membrane toward the spike zone, we're going to see the typical type of graded potential, and this resulting depolarization spreads to the spike zone, creating the receptor potential. And that receptor potential is the classical graded type that will decay over time and distance. And so the same phenomena apply when it reaches the spike zone. What impact will it have? Will it bring it to threshold or above threshold? And that receptor potential becomes a generator potential at the spike zone. Axon hillock is wrong for this case, this type of neuron, but still the spike zone, wherever it is, and this causes the firing of action potentials. Then, of course, you have an all or none phenomenon, as we're very familiar with. The same number of action potentials generated at the spike zone will be transduced in that pulse down the remainder of the conducting portion of this sensory cell. This illustration is more appropriate. Here we see the neuron cell body with the peripheral process, and we're recording the stimulus directly on the terminal, and we're seeing the graded potential, the receptor potential here, that's going to produce the generator potential near the trigger zone, which more appropriately is located here, way out on the peripheral process for a sensory cell of this type, especially for what we'd see with a stretch receptor on a muscle, as indicated in the previous slide. And then recording anywhere along the conducting portion of the fiber, whether it's in the peripheral process, the remaining portion that's myelinated, or the central process, we would see the action potential. So again, the graded potential is the receptor potential, and then the generator potential is what we would detect near the trigger zone, and how many action potentials would that produce. So the strength of the stimulus determines the amplitude of the receptor potential, and that impacts the amplitude ultimately then of the generator potential, the amplitude of the generator potential is directly correlated to the number of action potentials that will be generated at the spike or trigger zone, and thereby the firing rate, the frequency in a pulse is affected by the strength of stimulus, and we said that's correlated directly with how much neurotransmitter is released at the terminal of the central process of this sensory neuron. Thus, we can again discuss the coding of stimulus intensity. Here we are looking at a Pacinian corpuscle, and we can measure membrane potential at the sensory receptor, and then a voltometer close to after the spike zone on the conducting portion of the axon peripheral process will of course just show the action potential. Lower panel is showing the stimulus strength and the on and off, so a square pulse type of stimulus being delivered. And this middle panel shows and of course this is the x-axis is time in milliseconds, so over time we apply two stimuli with an interstimulus interval, and we notice that that becomes the interstimulus interval for measuring the receptor potential as well as the interpulse interval where we are measuring the action potentials. So the first stimulus of lower intensity produces a receptor potential that shows some evidence of decrement over time, and this when it reached the spike zone as a generator potential was still sufficient to generate 1, 2, 3, 4, 5, 6 action potentials in the same period of time with increased intensity and increased amplitude of the receptor potential and thus the generator potential. Then at the spike zone, the generator potential produced this number of action potentials that were conducted down the axon. We have 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have increased the firing rate of this neuron. So if these were delivered in this fashion, we see the frequency modulation that correlates with stimulus intensity, and we also see the interpulse interval that is directly correlated to the interstimulus interval. So we get that type of information encoded, ultimately back in the CNS where the neurotransmitter will be released from the terminal of the central process. Now we can look at the coding of stimulus for duration. So the previous figure was stimulus intensity. This now is stimulus duration. So again, we have our similar receptor with a stimulus onto that receptor, the Pacinian corpuscle being shown here. And so we are showing stimulus strength over time. So you note first in our two test panels that the stimulus intensity or strength is not altered. And even though we have only one model of receptor that looks very much like the Pacinian corpuscle, we are considering that to represent two separate populations generically, such that one is a slowly adapting receptor and one is a rapidly adapting receptor. The slowly adapting receptor, and again these are the same stimulus strength over the same duration of time, the slowly adapting receptor will sustain production of action potentials for the entire duration of the stimulus administration. For the rapidly adapting receptors, there will be a burst of firing at the initiation, of a slightly higher frequency than the burst that will occur at termination. So you can see that the rapidly adapting receptors indicate when the stimulus, and that will likely also be correlated with the intensity, but the frequency of firing will be rapid at the initiation. It will stop firing, and then when the stimulus is removed, there will be a few action potentials fired then. So we're sending a signal through the rapidly adapting receptors of on and off. So in axons associated with slowly adapting receptors, the action potentials are generated for the duration of the stimulus and generally decline slightly in frequency while the stimulus is persisting or persists. So you can see that we still get an indication in the slowly adapting receptor of initiation and termination. So the amount of neurotransmitter being released at the terminal of the central process of this sensory neuron will decrease over the duration of the stimulus. So still we're getting a similar signal of on and off, but we know we don't lose track of it, let's say, while it's on. Here we know when it came on, no longer paying attention to it. We know when it stopped. So the rapidly adapting receptors do not code for stimulus duration. They generate action potentials when the stimulus is first applied and when it ceases. So there's a question here. Can a stimulus of very strong intensity produce action potentials at such a fast rate that they sum? Explain this and why or why not. And of course the answer is here, and it is no, because action potentials have refractory periods. So there is a maximal firing rate for neurons dependent upon the refractory period, as was discussed in an earlier neurophysiology lecture. This figure is showing the coding of stimulus intensity by recruitment or populations coding. So we are now getting at a more sophisticated level. Rather than looking at a single neuron, we're getting at a more sophisticated level of looking at neighboring receptive fields and the recruitment of additional sensory neurons. So not only looking at one sensory neuron alone for the weak versus strong stimulus, but pools of sensory neurons that pick up similar stimuli in the weak stimulus versus the strong. So above we see the recruitment within a single sensory unit. The weak stimulus is only going to activate a certain subpopulation of the nerve terminals of the telodendria. So the depolarizing or receptor potentials radiating away from the sensory terminal will produce a certain level of graded potential at the spike zone. The more of those activated, of course, you're going to get the temporospatial convergence or summation, such that the generator potential would be of a higher or greater amplitude at the spike zone, and we already saw how that will impact the firing rate of this sensory neuron. So this is one way that the CNS can receive information on the strength of the stimulus. But it's rarely one neuron alone, so there are pools of neurons with neighboring or sometimes overlapping receptive fields, and it's the same. You may completely activate the receptor field that would look more like this on an individual cell basis, but the information coming to the CNS is different as far as pools of neurons in the CNS are concerned. In a strong stimulus, it's going to activate all three of the complete receptive fields of those sensory neurons. And again, you have to superimpose this type of phenomenon upon this. So all of this one might get activated, and only a portion of this one, and only a portion of this one. So in the CNS, we can get that unique localization, that the pinprick is exactly there, and it's deforming the skin on my fingertip, out to this extent, maybe only half of the way through that one. So this coding of stimulus intensity by recruitment or population codings is obviously the more sophisticated level of looking at it, but the more likely means by which our organisms make sense of this type of information. We now want to take another look at the responses of the slowly adapting receptors and the rapidly adapting receptors. What we are seeing here are panels A and panels B. In the upper figure, let's start with the lower graph of panel A, looking at slowly adapting receptors. We again see stimulus intensity on the y-axis and time on the x-axis. Note that there's 0 stimulus to start with, and then we proceed to a certain level. So in both experiments here, the slowly adapting and the rapidly adapting receptors, the duration and the amplitude or intensity of the stimulus are identical. In the upper graph, we are looking at receptor potential in millivolts, also, of course, over time, and it's not telling us the units of time here. Could be seconds or milliseconds. So in the slowly adapting receptors, we see that there is that decrement over time of the receptor potential. Therefore, the graded potential at the spike zone decreases over time. So there is a gradual decrease in the firing rate originating at the spike zone, as we had seen before. For the rapidly adapting receptors, we saw that there was an initial rapid firing rate with a termination, and then, well, with a disappearance of any action potentials, and then at the termination of the stimulus, there was a second set of action potentials of a lower frequency. And of course, the receptor potential that would become the graded potential at the spike zone reflects this. A higher amplitude at initiation for any one particular stimulus intensity being applied. That can occur only over a short period of time. So there's some physical property of the membrane that only allows it to occur then. And it is reset and then reactivated with the termination of the stimulus. So that overlapping of the receptive fields can become extremely important. Receptive field A and B are shown here with neurons A and B. So a stimulus of one intensity centered directly within receptive field A might not activate receptive field B unless it is a strong enough intensity to spread to the edges or the margins of the full receptive field for neuron A. If doing that, it will partially activate neuron B and provide that type of information to the CNS. This is a stimulus of certain strength or intensity, and it is spreading from the area innervated by neuron A to neuron B, but it has not completely filled receptive field B. So this begins to reveal the complexity of the nature of sensory receptive field integration. And it becomes much more complex than that, but we are investigating that. Now this becomes important when studying the dermatomes. There is an area of dermatomes where, say, right at the level of the nipple, it's the typical example used, stimulating just the nipple itself, is the fourth thoracic um, spinal segment. And as you move farther away from the nipple, it will begin, if you're moving toward the head cephalically, it would then proceed into an overlap zone where both T4 and T3 would be activated. Then you'd hit a zone where only T3 is activated, and then T3, of course, overlaps with T2, and so on and so forth. So the neurologist that is trying to localize exactly where the cutoff is for the sensory loss in a spinal cord injury must know those overlap and non-overlap receptive field zones quite well. Continuing with our overview of the central nervous system, we look at the organization, review CNS and PNS, the PNS containing the sensory division and the motor division for the information that we were just talking about, especially the sensory division of the PNS. But that is represented and reflected within the CNS, and of course motor output from our integrated circuit that we started with comes from the integration center, the CNS, and the output is through the motor division. The somatic nervous system or the autonomic nervous system, both is sympathetic and parasympathetic. Looking at the somatic first, of course, there are the cranial and the spinal nerves. There are 12 cranial nerves. And for the spinal nerves, there are 8 cervical nerves, 12 thoracic. We just talked about T4 innervating the nipple area, and T3 and T2 and T1 as we moved on up. Here we see cervical and thoracic, the upper, at least first thoracic nerve, contributing to the brachial plexus for the innervation of the upper extremity. So after the 12 thoracic nerves, then there's the 5 pairs, let's emphasize pairs, both right and left, of lumbar nerves, and 5 pairs of sacral nerves, and then just 1 pair of coccygeal nerves. And of course, the lumbosacral plexus emerges from the subset of the lumbosacral nerves to innervate the lower extremity. Well, all of these different types of sensory input, those that we examined from the skin very closely, the muscle spindle fiber and the joint kinesthetic receptor that we referred to and we'll look at in more detail, especially the muscle spindle and the Golgi tendon organ, are providing this sensory information that comes to the CNS. So we talked about receptive fields and receptor potentials and generator potentials and firing rates. Well, this is conveyed to the CNS, and local effects can occur with reflexes, as we will study in lecture 16. But also we know that information is going to ascend to various areas within the brainstem, and of course the brainstem is the site of connection of the cranial nerves, so the same thing would happen, local reflex, but also ascending information. And it might go directly to the cerebellum or to other areas from the cerebellum after synapse up to the thalamus, might have a synapse in the reticular formation of the brainstem, and another neuron thereby going to the thalamus, and then finally to the cortex. So we'll look at a few sensory pathways bringing information ultimately to the cortex, the highest level of function in the human organism and all organisms that have a cortex. And then of course the motor output is another aspect of that to modulate the the sensory reflex. Well, let's just then quickly review the classification of neurons as we begin to put together this whole circuit: sensory neurons, association neurons, motor neurons. So afferent, input, integration, and then efferent, or output. There's also the structural classification. Multipolar is what we're going to see a lot of now for the motor neurons, and the association or the interneurons of the integrating system, the CNS. The bipolar are rare sensory neurons, so that one illustration we saw is either some other species, definitely not humans. It was supposed to be a pseudo-unipolar neuron. I leave this always and to remind you that it's pseudo-unipolar, and this is the most common structural classification of sensory neurons that will be studied. Special senses, there will be bipolar neurons in humans. Other species might have more bipolar neurons. Less sophisticated species might have less sophisticated structures of their sensory neurons innervating their muscle tissue. But in humans, it's the pseudo-unipolar neuron with the dendrites getting the stimulus with the receptor potential, the generator potential on the peripheral process where the spike zone is, and then action potentials proceeding, being conducted all or none directly to the central terminals of the central process and dumping neurotransmitter on the CNS interneurons, most of which are going to be multipolar with many dendrites, multiple inputs onto it, local circuitry, input from the PNS, descending from higher centers, and so on and so forth. So very sophisticated integration of all that information to generate action potentials at the axon hillock spike zone for neurons and send that information to other areas. As well, this might be a motor neuron where the axon will then pass into the PNS from the CNS. It may even be somatic or visceral motor neuron, as we will see. So it's only specialized sensory areas such as the vestibulocochlear system and the retina and the olfactory system where these bipolar neurons will truly exist. So these will be chemoreceptors for taste and smell, either right at the nerve ending, which is the case for olfaction, but for taste we know that there's the taste buds that would interface with these nerve endings. For the vestibulocochlear, there are hair cells associated with these nerve endings. So the sensory neurons are specialized to respond to the specific stimulus, light, pressure, sound, temperature, pain, transmits information to other neurons in the brain and spinal cord. The inner neurons are found in the brain and the spinal cord. They receive the sensory input, transmit the information to another neuron or to groups of neurons. We now have to begin to picture, hundreds of thousands of neurons committed to certain functions distributed throughout the CNS. And then the motor neuron receives the information from the brain and spinal cord after it's been integrated and sends that to the muscles and glands to cause an appropriate response to the original sensory stimulus. So the receptor endings of the peripheral process of the pseudo-unipolar sensory neuron, also called the primary afferent neuron because it's the first one in the sensory circuit. The first interneuron to receive that information can be called the secondary sensory neuron or afferent neuron because it's still carrying beginning the integration of the sensory information coming in, conveys that through whatever circuitry there may be, and may be directly like this for a reflex, to the multipolar neuron that is the efferent neuron that will carry the signal to the effector. So the action potential, once it's generated by the spike zone in the peripheral process, of course, just is conducted all the way in to the CNS directly to the axon terminal, forming a typical exocytotic event for neurotransmitter release. Here we see a Golgi silver staining of a cortical multipolar neuron. Here's the soma with basal dendrites and apical dendrites very extensively and profusely branching. And somewhere down here on the basal part of the soma will be the axon that'll come out and leave. So extremely sophisticated structure, and thousands, probably more than 10,000 synaptic inputs to this neuron and its apical and basal dendrites. This is the pyramid-shaped soma there of this pyramidal cell. So tremendous amount of integration occurring on this type of sophisticated neuron. This classical illustration of a multipolar neuron doesn't do it justice at all. But still, we are viewing this as a peripheral nervous system containing the portion of the central process, but definitely the soma and all of the peripheral process with the receptors for the interface with the stimuli as we had described, and the central process entering the CNS, conveying the information, action potentials converted to correspondingly increasingly amounts of exocytosis and neurotransmitter release onto the second order neuron. So I called them primary afferent and secondary afferent. Now we see that it is an ordered sequence, and so we have the first order sensory neuron is the pseudounipolar. The second order sensory neuron is the one immediately postsynaptic to that. And then that typically will bring information to the thalamus as we make our ultimate goal to ascend within the CNS all the way to the cortex. And then the thalamus to the cortex will be the third order sensory neurons. So there's a variety of circuits for this, for the different modalities that we indicated with studying the different receptors. So there would be different names of the pathways to get to the thalamus, but ultimately this pathway from the thalamus to the cortex is the thalamocortical pathway. So we'll see that terminology as we proceed of naming these specific pathways. So the cortex is the ultimate target for conscious awareness. And a conscious awareness, of course, can occur even for visceral reflexes. We know when our urinary bladder is full and we need to urinate. We also know when our knee is bent or it's bent too far and it's painful. So the somatosensory cortex is shown here. We should recall the central sulcus here with the frontal cortex in front of it, the parietal cortex behind it. The gyrus in front of it is the precentral gyrus for primary motor function. The gyrus behind it is the postcentral gyrus, primary sensory cortex. We see the primary gustatory, primary auditory right nearby in the parietal cortex, and getting on to the temporal lobe down here, olfactory cortex in the temporal lobe. And of course the occipital lobe is basically committed to primary visual as well as secondary, tertiary. There's all these association areas after the initial sensory information is collected by neurons and then sent off to other interneurons for association and making sense of it all, assembling our world outside and inside into some type of representation within the nervous system. Here then, panel A and panel B, we see the classical illustration of the three neuron circuit, first order, second order, third order, for the ascension of sensory information all the way to the cortex. In panel A, we see the pathway called the dorsal column medial lemniscus pathway. In panel B, the spinal thalamic tract. So obviously we're going to have a neuron originating in the spinal cord with its axon ascending all the way to the thalamus for this tract. The dorsal column medial lemniscus makes us think that um, we're originating our second order neuron outside of the spinal cord, which indeed is the case. So the dorsal column medial lemniscus pathway shown in panel A transmits information from touch, pressure, vibration, and proprioceptors to the CNS. The spinothalamic is going to transmit information from temperature and pain. So this is temperature and pain, or pain and temperature is the typical way you hear it. It's also called the anterolateral pathway because the ascending spinothalamic fiber is in the anterior and lateral portion, so it's called the anterolateral pathway, spinothalamic, more common, I would say. So pain and temperature shown here, and proprioception and touch, vibration, and so forth in the dorsal column medial lemniscus. So stretch of a muscle, or the mechanoreceptor, as we had shown, the peripheral process carrying the information to the central process. We can have a branching with the synapse in the spinal cord, to provide local information on an event of a structure innervated by that level of the spinal cord. But to get that information to the higher centers, especially the cortex, a collateral of this central process ascends a great distance all the way to the brainstem, the medulla oblongata, and forms a synapse on the second order sensory neuron. So this was all first order sensory neuron here. The synapse between the first and the second order sensory neuron occurs in the brainstem outside of the spinal cord. So these are obviously very long axonal pathways, and they're always heavily myelinated and very rapid conducting systems. The second order sensory neuron originates in the dorsal column nuclei, as they are called. So they're in the dorsal, or the backside columns of cell bodies. So dorsal columns. And then there is a ribbon-like band of the second order sensory neuron axons. So meniscus means ribbon, and it's the one located more medially, so obviously there's a lateral meniscus for something else. So the medial lemniscus, so dorsal column, which is also the term for the white fiber tract ascending in the spinal cord, the dorsal columns. So the dorsal column, referring to the first order sensory neuron, synapse in the dorsal column nuclei, and then the second order sensory neuron pathway through the medial lemniscus, crossing the midline. All of these sensory and motor functions are crossed, you know that. Right brain gets left body, crosses in the medulla, and then ascends to the thalamus for synapse with the third order sensory neuron that is in the thalamocortical pathway that goes to the post-central gyrus, the primary somatosensory cortex that we had indicated in the previous slide. For pain and temperature in the spinothalamic pathway, the nociceptor thermoreceptors are the first order sensory neurons, the central process synapses at the level of the spinal cord where it enters, and the second order sensory neurons are in the gray matter at that level, and the axons of the second order sensory neurons cross immediately and ascend in the anterolateral pathway all the way up to the thalamus, synapsing on the third order sensory neuron, and the third order sensory neuron with the thalamocortical pathway also terminates in the primary somatosensory cortex. There are a lot of neurons in that post-central gyrus, so plenty of room for all of these afferents to come in and innervate it. We look at these in a little bit more detail at higher magnification in these next couple slides. So proprioceptors, the first order sensory neuron, again, some local information for reflex action within the spinal cord, ascend in the dorsal columns to synapse of the second order, always at a point of decussation, so the dorsal column medial lemniscus crosses or decussates in the the medulla oblongata, or the brain stem. Still, thalamocortical is the third order sensory neuron. So we see in these two examples, it is the second order sensory neuron whose axon crosses the midline, but for the distinction between the two, the spinothalamic pain and temperature crosses at the level of the entry of the first order sensory neuron in the spinal cord itself. So the spinothalamic decussation is right within the spinal cord, and still the third order sensory neuron is thalamocortical. So we saw with the dorsal column's medial lemniscus pathway that there was a collateral that branched from the central process of this primary afferent, or first order sensory neuron, most of which is in the PNS, but the central process enters the CNS. So that decussation allowed for synapse on the second order sensory neuron in the dorsal column nuclei to form the medial lemniscus that ascended to synapse in the thalamus, the third order sensory neuron. The other collateral we saw was forming a terminal right in the spinal cord at the level of entry. It can synapse with an interneuron there and contribute to the reflex for a more immediate and locally integrated output response. We know that ultimately the second order medial lemniscus and then the third order thalamocortical is going to activate descending motor that comes down and impacts this circuitry to modulate it. So if you are putting conscious effort into focusing on a reflex, you can accentuate it or inhibit it, blunt it, or attenuate it due to the cortical input. So this ends our discussion of the sensory receptors and the integration between PNS and CNS of this sensory